whether I'm audible and video is visible. 1 minute. Yeah, I'm launching the poll. Kindly tell me whether I'm audible and video is visible. Okay. Okay. Okay, fine. Let us start the class, guys. I will share the PPT. Okay, fine. 1 minute. Okay, I think I will off the video. 1 minute. Okay, fine, guys. So, guys, in the today's class, we are going to talk about the biomolecules chapter. Okay, biomolecules chapter. From the biomolecules chapter, you are going to get around 2 questions. Okay, kindly remember that. Okay, fine. Let us talk about the biomolecules. If my audio and if anything is audio, you are getting the breaking of voice means kindly tell me in the chat box. Okay, fine. The biomolecules, mainly we study about the biomolecule. All the organisms is made up of the molecules that we will call as a biomolecule. Okay. Let us talk about this, guys. Okay. All the organic molecule, okay, all the organic molecule which is made up of the organism is called as a biomolecule. And also we will take that as the food also. For example, you can take a carbohydrate, protein, carbohydrate. Here you can see carbohydrate, protein, lipids, and nucleic acid. These all the things you will take in the food also, right? So that is all nothing but the biomolecules. Each of the molecule in your or organism is made up of these all the molecules. Okay, fine. In the biomolecules, mainly we will see the two types of the molecules, right, guys. Okay, one is a macromolecule, and another one is the micromolecule. Micromolecule. What do you mean by macro? Macro means you all know that it is a big, right? Big in size. Micro means what? That is a smaller in size. Okay, smaller in size. Okay. Then coming to the macromolecule. Macromolecule is having the molecular weight more than 1000 Dalton. Okay, more than 1000 Dalton is called as a macromolecule. The example for the macromolecules are in the carbohydrate, polysaccharide like cellulose, starch. These all the things will come under the polysaccharide, right? Polysaccharides and proteins. Proteins are nothing but you can take the carbohydrates and proteins and lipids and also the nucleic acid. Nucleic acid, which are those? DNA and RNA. RNA. Will all these comes under the macromolecule. Okay. This all comes under the macromolecules, and these macromolecules are not soluble in trichloroacetic acid. Trichloroacetic acid. Remember, these are insoluble. Insoluble. Okay. Macromolecules are insoluble in trichloroacetic acid. These are more than 1000 kilodalton. The examples are polysaccharide or carbohydrate, protein, lipids, nucleic acids. Okay. Then coming to the micromolecules. Micromolecules are lesser than 1000 kilodalton. 1000 dalton. Okay. These are lesser. Okay. Lesser than 1000 dalton. And the example for these are amino acid. Okay. Then glucose, These all are the single molecules, right? Glucose, fructose, these all the things will comes under the micromolecules, and also the nucleotides. Nucleotides. Okay. These all the things will comes under the micromolecule. Okay. Remember this. Okay. Then coming to the metabolites, the primary metabolites and the secondary metabolites. What do you mean by primary metabolites? Is these are mainly, primary means what? Primarily, okay. These are primarily required for the organism for the development and for the growth and for the reproduction. So that's why it is called as a primary metabolites. Okay. These primary metabolites are, we will call it as the, these are required for the normal physiological functions. Okay. What are these example for the primary metabolites? What we will take mainly as the food, that all are the carbohydrate, protein, nucleic acids, and the lipids. These all are important for the normal physiological growth. Okay. Then coming to the secondary metabolites. Primary metabolites are produced by all the organisms. Okay. Then coming to the secondary metabolites are mainly produced by the plants and fungi. Okay. Mainly plants, 1 minute. Okay. Mainly plants and fungi will produce these secondary metabolites, and these are not required. These are not having any role in the physiological function, but they required in the immune system, means defense. These are required for the defense purposes. Okay. The examples are carotenoids, morphine. We will study in detail about the secondary metabolites here. Remember, primary metabolites required for the normal physiological function, whereas secondary is not required for the normal physiological function, but these are produced by plants and fungi. The animals will not produce this. Okay. Fine. Let us go to the secondary metabolites. Okay. So here, this slide is very, very important, and you will get the question from this in the match the following type. Okay. And also in the statement wise question. Kindly remember the examples here. Coming to the pigments, guys, you have seen that vegetables are in the different colors, right? For example, if you take the brinjal, it is in the purple color, and coming to the capsicum, yellow color, red color, you can see, right? In the pigments, mainly the carotenoids, the orange color of the carrot, and yellow color, you can see the capsicum. It is mainly because of the carotenoids. And anthocyanin, the purple color of the brinjal, is due to the anthocyanin. Remember, in the pigments, carotenoids and the anthocyanin, alkalides, these are usually the drugs. Usually, the alkalides will come, usually the morphine and codeine is used. Those all are the morphine and codeine. Remember, alkalides are morphine and codeine. Then coming to the terpenoids, the plant hormone is there, made up of the terpenoids, monoterpene and diterpene. How can you remember terpenoids, guys? This is also end with the terpenes only. Terpene. Okay, remember. And essential oil is a lemongrass oil. Here you can see this lemongrass oil is mainly used for preparation of the tea, some green teas, and also for the oils they will prepare. And coming to the toxin, you have to remember the toxins. Abrine and ricin are the toxin. And coming to the lectin, lectin is the carbohydrate binding protein. Okay. So the example is concavalin A. Concavalin A comes under the lectin. Remember this, precious. It is very important. Okay. Then coming to the drugs. In the drugs, vinblastine and curcumin is used as a drug. Vinblastine is mainly used in the cancer treatment. Cancer. Okay. Then coming to the polymeric substances, you already know rubber, gum, and cellulose will comes under the polymeric substances. Kindly see this slide once again. This is very, very, very important. Once again, I will tell you, in the pigment, carotenoid and anthocyanin. In the alkalides, morphine and codeine. Terpenoids, how can you remember? Terpene, it will end with the terpene, and alkalides will end with the ene, ine, okay. And coming to the toxin, abrine and ricin, two will come. Lectin will be concavalin, and drugs, vinblastine and curcumin, and polymeric substance, rubber, gum, and cellulose. Okay. Is that okay, guys? Did you understood this secondary metabolites? Can you give me thumbs up if you understood means? Okay, fine. Then let us go to the question, guys. The first question is, which of the following is not a secondary metabolite? Secondary metabolite. Okay. Option A, curcumin, and option B, morphine, option C, anthocyanin, and option D, lecithin. You can answer me in the poll also. Okay, I will launch the poll. You can answer me in the poll. I'm launching the poll. You can answer me, guys. 1 minute, 1 minute. Sorry. Sorry. Yeah, MCQ poll. Yeah, I launched it, guys. You can answer me. In the chat box also, you can answer me. Think once again. Think once again. Which one we didn't study here? Very good, guys. Very good. Okay, so I will end the poll. So many of them given the correct answer. I'm sharing the result here. Yeah, option C by 33% and option D by 67%. Okay, no issue. Kindly see here. We have studied this, right? Option D is the correct answer, guys. See here. We studied the curcumin. That is as a, here you can see where is the curcumin? In the drugs. We have studied the drugs. And then coming to the morphine. Morphine is the alkaloid. Anthocyanin is a pigment, guys. Now only we studied, right? Brinjol. Brinjol is made up of anthocyanin, the purplish color. Because of the pigment anthocyanin. Anthocyanin is a secondary metabolite. Then coming to the lecithin. Lecithin is a phospholipid. Phospholipid. Where you have seen the phospholipid, guys? On the plasma membrane. Right? Lecithin. It is nothing but phospholipid. Okay. The correct answer is option D. Lecithin will not come under the secondary metabolite. And here is another question. Match the list 1 with the list 2. Here the list they are given. Here they are given the example. You have to match that, guys. Okay. I will launch the poll. You can again go through the poll. Okay, I'm launching the poll. You can go through the poll, guys. What you have to do? You have to match. Right? First thing what you have to do is the matching. Okay. Nukraju is given. Okay, then what about others? First, you know what are the things you know, guys. First, match with that. Okay. I will answer to this. You can go through that. Terpenoids. Which is the example for terpenoids, guys? Terpenoid. It is also end with a pain only, right? Diterpene. For A, it is 2, right? For A, it is 2. Okay. Then coming to the lectin, you know aware of lectin, right? Lectin is the concavalent, right? Then match with this. A should be have the 2. A should have the 2. Everywhere A is having the 2. Then coming to the lectin, B is having the 4. Where is the B is having 4? In both the A option and B option. Then we'll remove these 2. Okay. Then coming to the alkaloid, we know about the alkaloid. Which is example for the alkaloid, guys? Codeine, right? Then coming to the toxin, yes. The correct answer is option D. Right. Here you can go through this. Yes, option D is the correct answer, Siman. Yes, Mahima. You'll get the confusion, right? We'll also get confusion. So kindly see this once again, okay? I'm sharing the result. 33% given C and 67% given D. Okay. So here how they are matched. Here B is having the and C is having the alkaloid is the codeine, right? And toxin is nothing but the abscine and ricin, right? Okay. Fine, guys. Let us go to the carbohydrates. Carbohydrates, you all know that carbohydrate is made up of carbon, hydrogen, and oxygen, right? And the formula is, you can take the glucose. The formula is C6H12O6, right? And you can see here, uh, hydrogen and water combination, here you can see that ratio is as the H2O only, right? It is looks like H2O only, right? The same thing happen in the water. So that's why it is called as the hydrates of carbon. So that's why the name is carbohydrates. Hydrates of carbon, right? Okay. And what is the carbohydrates actually? Carbohydrates are polyhydroxyaldehyde and polyhydroxy ketones. Here you can see It is mainly consisting of the so many hydroxyl group, right? So that's why it is polyhydroxy, and it is having the aldehyde group in the glucose, and it is having the ketone group in the fructose. So that's why the carbohydrate is called as polyhydroxyaldehyde and polyhydroxy ketone. Okay. And you know that carbohydrates are soluble in water, except polysaccharide. Okay. Fine. They are the macromolecules. These are the macromolecules, carbohydrate. And coming to the carbohydrate, the division of the carbohydrate, mainly the carbohydrates are divided into simple and complex. In the simple, there is a monosaccharide and oligosaccharide. In the oligosaccharide, disaccharide we are going to study, and polysaccharide. Monosaccharide means what, guys? Single. Single. Mono means single. Okay. It is consisting of the single sugar. Saccharide means sugar. Okay. Single sugars it is made up of. The example for the monosaccharides are glucose, fructose, galactose. Can anyone tell me any other examples, guys? Can anyone tell me the examples of any other things? Yes, ma, I will add your number. Okay. You are given your number, right? I will add you in the group. Okay. Thank you, pa. I will add surely. Okay. Fine. Can anyone tell me, guys, other than this, any examples of the monosaccharides are there, guys? Monosaccharide examples? I need your answer in the chat box. Mannose. Mannose will come under the monosaccharide only, right? Mannose. Mannose. Okay. The monosaccharides are glucose, fructose, galactose, and mannose. Don't confuse with the lactose. Okay. Then coming to the disaccharide, di means two. It is made up of two sugars. Examples are maltose. Maltose is made up of glucose plus glucose. How it is linked, guys? Two sugars are linked by the glycosidic bond. Glycosidic bond. Remember, okay? So disaccharides are made up of maltose. Maltose is made up of glucose and glucose, and lactose. Lactose is made up of galactose. Galactose plus glucose, and sucrose is made up of fructose and glucose. Remember, fructose and glucose, sugar. Okay. And you know that sucrose is called as non-reducing. Sucrose is called as non-reducing sugar. Why we are calling as a non-reducing sugar? You can see here. This is not having the reducing end. It does not have the reducing end. That's why it is called as a non-reducing sugar. Okay. Then coming to the polysaccharide. In the polysaccharide, mainly two types. One is homopolysaccharide and heteropolysaccharide. Homopolysaccharide means it is made up of the similar unit. Okay. It is made up of the similar unit. So that's why it is called as a homopolysaccharide. For example, starch. Starch is made up of the only glucose. Only glucose. Okay. Then glycogen is made up of only glucose. Starch is acting as a storage polysaccharide in the plants. Plants, right, guys? Then coming to the glycogen. Glycogen is acting as a storage molecule in the animals. Where is the glycogen is going to be stored, guys? In the liver. Remember. And coming to the inulin. Inulin is consisting of the fructose. Fructose. Okay. Then coming to the cellulose and chitin is a structural molecule. Cellulose is also made up of only the glucose, and chitin is made up of N-acetyl glucosamine. We have seen in the cockroach, the arthropodial exoskeleton is made up of chitin, is made up of a homopolysaccharide. It is nothing but the N-acetyl glycosamine. Okay. Then coming to the heteropolysaccharide, it is made up of the two or more repeating units. Repeating units. That's why it is called as hetero. And example is peptidoglycan in the bacterial cell wall, and it is made up of NAG and NAM. NAG plus NAM. N-acetyl glucosamine and N-acetyl muramic acid. Did you understood the carbohydrate, guys? This slide, have you understood? Did you understood this slide? Yes, Shashank, I will add that also, pa. Very good, Sai Charan. Yes. Okay, Sai Charan. Okay, fine. I think you understood this means, can you give me thumbs up, guys? Okay, and here the mainly thing you have to remember here is the starch. Starch is mainly made up of amylose and amylopectin. Amylose is not branched. It is not branched. Okay. Then coming to the amylopectin, it is having the branched structure, guys. Okay. And coming to the glycogen, glycogen is also branched structure. And cellulose is not branched. It is not branched. Okay, remember this. And coming to the inulin, it is made up of fructose. And glycogen, you can see, guys, the left end is a non-reducing and right end is having the reducing end. Okay. In the exam, they may ask you like this. The glycogen is made up of around 200 molecule of the glucose. And how many reducing end are there? Only at the end, one glucose should be there, right? It is having only one reducing end. You have to remember that. Okay, only one reducing end. No matter how many glucose is there, only one end is there. So that's why it is having the only one reducing end. Okay. And coming to the starch, starch will give the positive for the iodine test because it will hold the iodine molecule in the helical structure. Whereas the cellulose will not give because the cellulose is not having the helical structure. So that's why it will not give the positive for the iodine test. Okay, remember that. And chitin we already studied. This is about the polysaccharide, guys. And polysaccharides are not sugars. Okay, they are not sweet in taste. Whereas the monosaccharides are sweet in taste. Okay, sucrose is also sweet in taste. Polysaccharides are not soluble in water also. Whereas the monosaccharide is soluble in water. Okay, so we have completed with the structure. We have completed with the carbohydrate. Then we'll go to the protein, guys. Okay, where you have seen the protein, guys? In the egg, milk, right? In the pulses. Every day we will eat the protein, right? Protein is the main component that is made up of the body, right? The enzymes and all the thing is made up of the proteins only. We'll study about the protein. The proteins are mainly made up of, we will call as a protein as the polypeptides, guys. Remember, important. Proteins is important. Okay. Proteins are made up, we will call it as a polypeptides. And the mainly molecule made up of the proteins are amino acids. For example, if you take a building, each building is made up of the brick, right? Like that, the each polypeptide or each protein is made up of the amino acids. Okay. And they are linear chain of the amino acid. And we will call it as the polypeptide. And these are not homopolymer. Okay. These are heteropolymer. Heteropolymer. Okay. Hetero. Okay, remember this. And we can see there are the 20 standard amino acids are there. And we can see the essential amino acid, which cannot be synthesized in the body. So that's why we are calling it essential. And the non-essential amino acid are already synthesized in the body. For example, you can take when you are writing the things required for the home ration. Okay. Are you going to write already there in the home? Which are the ration already there in the home? For example, rice and oil is there already there in the home. Are you going to write that also in the ration list? No, right? Those are non-essential. Like that, non-essential amino acid already synthesized in the body. Those are not required. Essentials are required. Okay, we'll study which are the essential amino acid and non-essential. Okay, and this is the amino acid structure, guys. The amino acid, why we are calling it the amino and acid means it is consisting of the amino group and also the acid group. Okay. And those amino and acid is attached to the alpha carbon atom. And this is having the R group, that is a side chain. Okay. Remember this. And we all called as polypeptide. In the protein, we can see the bond that is the peptide bond. And also peptide bond is also called as the amide bond and the CN bond. CN bond. Okay. Remember, peptide, amide and the CN bond is all are the same. Okay. For example, now here they are given the structure, guys. You have to remember these amino acid, the glycine. See here, amino group is there, acid group is there, and the R group is simple. That is the hydrogen. Okay. R group is very simple here. Glycine. Glycine consisting of the hydrogen. Then coming to the alanine. Alanine is having the methyl group as the R group. Okay. Then coming to the serine. Serine is having the hydroxyl as the R group. Okay. Remember this structure. It is very important. They will give this diagram and they may tell you to identify the which structure it is. Then coming to the tetrapeptide. In the tetrapeptide, there are the 4 amino acid is made up of. Okay, and here you can see these all are the amide bond or the peptide bond. Okay, C double bond O, NH is there. So that is nothing but the peptide bond. Okay, if the 4 amino acid is there, means we will call as a tetrapeptide. Tetrapeptide. But here we are having only the 3 peptide bond. Only 3 peptide bond. 1, 2, 3. 3 peptide bonds are there. Okay. If it is the 5 amino acids are connected, can you tell me how many peptide bond will be there, guys? If the 5 amino acids are there, means how many peptide bond will be there? Can you tell me how many peptide bond will be there if the 5 amino acid means? Very good. 4. If the 6 means 5, right? Minus 1 will be the peptide bond. You have to remember. Okay. Fine. Remember the peptide bond also called as amide bond and CN bond. CN bond. Okay. Fine. Okay. When these amino acids are in the water, when this amino acid in the solution type, it will be exist in this Zwitter ion. What do you mean by Zwitter ion, guys? Zwitter ion means it is having both positive and the negative charge. Okay, it is having both positive and negative charge. So that's why it is called as a Zwitter ion. Okay, remember this. Then coming to the some of the important amino acid you have to remember, guys. Around the in the 20 amino acids, you have to remember these amino acids. Okay. First one we will go to the acidic amino acid. Acidic amino acid. There are two types. One is the glutamic acid and aspartic acid. Why we are going to call as acidic means you see here in the amino acid, acidic group also there, amino group also there, right? If the amino acid contain more than one acidic group, see here, another acidic group is there, right? So that's why it is called as the acidic amino acid. Here also you can see one more acidic group is there, one more acidic group is there, right? So these all acidic amino acid, glutamic acid and aspartic acid. Then coming to the basic amino acid, basic consisting of the one more amino group. Amino group will be extra here. Okay, amino group. See here, the mainly basic here, amino group is there here, right? One carboxyl group is here, there, right? Here you can see more amino group is there. That's why it is called as basic amino acid. The example for the basic amino acids are histidine, arginine, and lysine. Remember this. Okay. Then coming to the aromatic amino acid, these aromatic amino acids are having the benzene ring. You can see this benzene ring. Example for the aromatic amino acids are phenylalanine, tyrosine, and tryptophan. How can you differentiate this? All the thing means the phenylalanine does not have the hydroxyl group. Only benzene ring is there. Okay. Whereas in the tyrosine, the benzene ring is there along with OH group. Okay. The tryptophan is having the 2 ring. Remember like this. Okay. Fine. Then coming to the essential amino acid, you can go through this PPT, guys. Here it is having the essential and non-essential amino acid. Whereas in the adults, histidine and arginine, okay, histidine and arginine is called as semi-essential. Semi-essential. Sometime it will be produced or sometime it will be not produced. So that's why it is called as semi-essential. Histidine and arginine. Remember this. Okay. Fine. Then coming to the structures of the protein. I think you already studied this in your chemistry chapters also. We'll study once again. There are 4 structures are there. Primary, secondary, tertiary and quaternary. What do you mean by primary, guys? If you take the charger wire, so the amino acids are, you can see here, the amino acids are linked like this, right? Amino acids are linked like this. 1, 2, 3, 4, 5 amino acids are there, right? Only between this, the peptide bond we can see, right? Peptide bond we can see. So this is called as a primary structure. So what is this primary structure? If I told you people to stand up in the line, okay, holding the hand, you will stand by holding the hand, right? So that is nothing but we will call it as the primary structure. For example, in the primary structure, you can see the first amino acid is having the amino group. Okay. The first amino acid having the amino group and last amino acid is having the carboxyl group. Okay. Remember this. Okay. This is a primary structure. Primary structure we can see only in the peptide bond. Then coming to the secondary structure. In the secondary structure, what will happen? The same single polypeptide only there, but the single polypeptide is that is RN that is having the spiral-like structure. Spiral-like structure. Okay, that is called as the secondary structure. For example, you can see here I have taken this, right. Now this only having the spiral-like structure. This is a secondary structure. In the secondary, we can see alpha helix and beta sheet. In the beta sheet, it is arranged like the book sheet. Like the book, how can you see the book sheets will be right? Like that, the secondary structure you can see. Okay. Okay, 1 minute. Huh. In the alpha helix, the example is the keratin. Mainly, they will give the protective or the mechanical strength. Okay, the secondary structure. Mainly, you can see that in the hair. In the hair, you can see the keratin will be there, right? The keratin is having the alpha helix structure. And beta pleated sheet, you can see in the silk. Silk. You have seen the silk sari and silk dhoti, right? In that, mainly the silk is made up of the beta pleated sheet. Beta. The alpha is seen in keratin in the hair. Okay. Then coming to the tertiary structure, the same single peptide only. Single peptide only. The if you take the thread and if you see the woolen color, woolen that is made up of the pink color. If you take means, if you wind up that means what it will look like, a wind up structure it will look like, right? You can see this like kind of things, right? That is made up of single only, single peptide only. Okay, but it is holding, right? The structure is holding, right? That tertiary structure. In the mainly tertiary structure, you can see the so many bonds. Okay, one is a non-covalent bond, non-covalent, and the covalent bonds. You can see a non-covalent bond. Can you recall, guys, which are the non-covalent bonds? Which are the non-covalent bond, guys? Can you recall? Van der Waals force of attraction, electrostatic force of electrostatic force, right? Then non-covalent bond, hydrogen bond, right? These all the things will comes under the non-covalent bond, and covalent bond is the strongest bond, right? These all the things are present in the tertiary structure, whereas the secondary structure and primary structure is not having. Okay, and the hydrogen bond. In the secondary structure, you can see the peptide bond and hydrogen bond. Okay. Whereas in the tertiary structure, you can see all the types of bonds are there. Okay. Non-covalent and covalent bond are there and made up of only single peptide. Single peptide. The example for the tertiary structure is enzymes. All the enzyme or biological activity require the tertiary structure, and myoglobin. Myoglobin is present in the muscle, right? It will store the oxygen. That is mainly having the tertiary structure. Example for the tertiary structure is myoglobin. Myoglobin is having only the single peptide. Okay. Then coming to the quaternary structure, the quaternary structure is having more than one polypeptide, one polypeptide. So that's why it is called as quaternary structure. The example is hemoglobin. Hemoglobin is having the quaternary structure that is having the 2 alpha and 2 beta, 2 alpha and 2 beta. Remember, okay? All the biological activity of the protein require the tertiary structure. Okay. For example, if you taken some protein and if you boil that, you are given the heat. Okay. What will happen? This all the bond is going to be broken down, right? All the bond is going to be broken down. Only it will be with the peptide bond. That is the tertiary primary structure. It will be there. Okay. So that's why we should not heat the protein more. Or that's why the enzymes will lose its activity when we given the high temperature, right? Because it will lose its tertiary structure. That's why. Okay. So here are the examples, guys. Collagen, in the last class we have studied, right? It is made up of the intracellular ground substance. Collagen, you can see in the skin. And trypsin. Trypsin is the enzyme that will break down, that will broken down the protein. Then insulin, you know that insulin, it will, it will take the, it will take the glucose that is present in the blood and it will synthesize the glycogen, right? It will synthesize the glycogen from the sugar that is present in the blood, right? It will decrease the concentration of the glucose in the blood, insulin. So that's why diabetic patients will take the insulin, right? That is a hormone. Then antibody, you know that antibody will fight against the infections, right? Then receptor. If you are sleeping in the room and mother is doing the biryani or any favorite palav or anything, you will get the smell, right? How can you get the smell? The receptor is present in your nose, right? And if any dish is very tasty, how can you tell that very tasty? The receptor is present in the tongue, right? Those are the receptor made up of protein. Then coming to the GLUT4. GLUT4 enables the glucose transport into the cell. Glucose is present in the blood that should reach the cell, right? That is mainly by the GLUT4. Remember, okay? And the abundant protein in the animal world is a collagen. That is made up of, mainly the skin is made up of collagen, right? That is abundant in the animal world. In the world, the abundant protein is rubisco enzyme that is present in the plants. Okay, so let us solve some of the questions, guys. So here is the MCQ question. I think you are aware about the proteins now. Now you can solve this question. Read this question first. Then I will on the poll. Here it is also MCQ based question. What you had to do? You had to match any one first. Okay, you know the trypsin. What is the trypsin? You know what is the insulin now only, right? What is the collagen? Match this with very good Shruti, very good Sai Charan. Now you match this. Now I will run the poll, guys. I will on the poll. You can answer me in the poll. Okay. You can answer me in the poll also. You can answer me. Athini, very good. Krupa, Nurka, okay. We will match it, guys, here only. Okay, we will match it here. So what is the GLUT4, guys? GLUT4 enables the glucose transport into the cell, right? Then A should have the 4. Okay. Only in the option A, it is having the 4. Right? Again, we will recheck. Okay. What is the insulin, guys? Insulin is a hormone. Then B should have the 1. Right? Then coming to the trypsin. What is the trypsin? It is the enzyme that will break down the protein. Right? What is that? C. C is the 2. That is also there. And the collagen should be 3. Intercellular ground substance 3. Which is the correct answer, guys? Option A. Right? So many have given option D in the chat box. I'm ending the poll. Very good. But everyone given the option A only. Very good, guys. Okay. Okay, then we'll go study about the nucleic acids, guys. What is this nucleic acids? These are the macromolecules. Nucleic acids are the 1 minute. Okay. So nucleic acids are the macromolecules. You all know that nucleic acid nothing but the DNA and RNA. It is helpful in the transferring the genetic information from the parents to the offspring, right? So mainly the nucleic acid, DNA and RNA is made up of the nucleotides, not sites. Kindly remember tides, okay? Tide. We use for the washing, right? Tides, okay? Mainly it is made up of nitrogen base, pentose sugar and phosphate group. 3 things are there. Nitrogen base, pentose sugar and phosphate groups are there. Okay. In the nucleoside, only a nitrogen base and pentose sugar is there, guys. Okay. If they ask the example for the nucleotides, the tides usually nucleotides are having the acidic group. Acid. We will end with the acid. Examples are adenylic acid, thymidinic acid, guanylic acid, uridylic acid, and cytidilic acid. These all are the nucleotide, end with the acid. Acid, remember. Then coming to the nucleoside, it will end with the sign. Okay. Adenosine, guanosine, thymidine, uridine, cytidine. These are nucleosides. Okay. Then coming to the what is the nitrogen base, you all know that the nitrogen is made up of a yes, that is a double ring, that is a purine. That purine examples are the adenine and guanine, and the pyrimidine. Pyrimidine are having a single ring, that is nothing but the pyrimidine are cytosine, thymine, and the uracil. Right? These all the things will comes under the pyrimidine. Remember, purines are guanine and adenine, and pyrimidines are cytosine, thymine, and uracil. Then coming to the pentose sugar, guys. Pentose sugar, the DNA is having the deoxyribose, does not contain the oxygen. You see here, deoxyribose. In the second carbon, it is not having the oxygen. So that's why it is deoxyribose. Then coming to the RNA, that is having the, that is a ribose, means it is having the oxygen at the second carbon. Okay, remember that. Then what the thing you have to remember is the DNA. DNA mainly consisting of purine, that is adenine and guanine is there, purine, whereas the pyrimidine, it is having the thymine. Thymine and what is another thing? Cytosine. Thymine and cytosine is there. Whereas coming to the RNA is having the adenine and guanine, and it is having the uracil and cytosine. Uracil and cytosine. Thymine is absent in RNA. Okay. The adenine and thymine pair up with a 2 hydrogen bond. Guanine and cytosine bond up with 3 hydrogen bond. Okay. 2 hydrogen bond and here 3 hydrogen bond. Remember. Okay. This is about the nucleic acid, guys. Then fine, coming to the lipids. And one more thing, guys. Here you have to remember the nitrogen base attached to the sugar by the N-glycosidic bond. N-glycosidic bond. Okay, remember. Fine. Then coming to the lipids, guys. Lipids are water insoluble. You can see, you can take a 1 cup of water and put the droplet of the oil. It will not be soluble, right? So lipids are insoluble in water. The lipids, if you take the lipids, guys, lipids, it is mainly made up of fatty acids. Okay, fatty acid. These fatty acids are attached to the glycerol. Fatty acids are attached to the glycerol. We'll talk about the examples of fatty acid. There are saturated fatty acids or unsaturated fatty acids. What do you mean by saturated fatty acid? You all are happily eat the chocolate, ice cream, biscuit, and all the thing, right? These all are made up of saturated fatty acid. These all are not good for the health. Okay, not good for the health. That is mainly made up of single bond between the carbon. Okay, and vanaspati ghee. These all are saturated fatty acids are solid at the room temperature. Example, vanaspati ghee, dalda, these all the things will come that is saturated. These all are solid at the room temperature. Then unsaturated fatty acids are liquid at the room temperature. These are mainly oils. Okay. So here you can see there is a double bond. Double bonds are there in the unsaturated fatty acid. These all are good for the health. Okay, fine. And you can see, guys, fatty acids, the examples are saturated and unsaturated. The glycerol, you can see the structure of the glycerol here. It is very important. For the glycerol only, we are going to add the fatty acids. Okay, glycerol structure is there here. For the glycerol, we are going to add the fatty acid. So then it will become the triglycerides. Okay, so triglycerides, and here you can see the phospholipid structure. It is also called as a lecithin. It is mainly made up of the plasma membrane. Okay, and you can see fatty acids are called as the amphipathic in nature. Amphipathic in nature means it is having both hydrophilic head, hydrophilic head is there. Okay, and hydrophobic tail is there. Hydrophobic tail is hydrophobic, remember. Okay, and you can see that gingery oil having a low melting point and phospholipid that is found in the lecithin. And you have to remember the cholesterol structure, guys. Mainly the lipids are made up of um, simple lipid. In the simple lipid, the oils and fat will come. Compound lipid. In the compound lipid, phospholipid will come. Compound lipid. Okay. Compound lipid will come. And in the esterified lipid, here the cholesterol will comes under the esterified. Here you can see cholesterol on the third carbon. Third carbon, it is having the OH group. Okay, third carbon. Remember, cholesterol are very important for the synthesis of the hormones, right? In the female, we can see the estrogen, right? Estrogen, this all the hormone is made up of the cholesterol only. And in the male, testosterone hormone is made up of the cholesterol only. Okay, fine. Then coming to the nature of the bond, in the protein, we can see the peptide bond, and in the carbohydrate, we can see the glycosidic bond, and then the nucleic acid. Mainly between the two nucleotides, we can see the phosphodiester bond. Okay, phosphodiester bond, and you can see that between the sugar and the here you can see the phosphate group is there, and The ester bond is present between the sugar and the phosphate group here. And the DNA structure, you can see the phosphodiester bond, glycosidic in the carbohydrate. Okay. And mainly in the lipids, you can see the ester bonds. Ester bond. Okay. Fine. And this is about we study about the nucleic acids and the lipids and also protein and the carbohydrate. Now we'll come to the very important point, that is the enzymes. Okay. Guys, I think you are aware of the catalyst, right? In the chemistry lab, you will take the catalyst for increasing the reactions, right? The same, the enzymes are called as biocatalyst. Biocatalyst. This biocatalyst is present in the living organism. Okay. This almost all the enzymes are protein only, guys, and these are made up of tertiary structure. Remember, okay? Tertiary structure it is made up of, and only the one enzyme, that is a ribozyme, is having the RNA. Remember, ribozymes are having the RNA. Okay. And you know, this is the enzyme. Examples, the enzymes are looks like the protein, guys. That is having the structure where the substrate is going to bind, right? Here you can see this is the enzyme. The substrate is going to come and bind to the enzyme place. That is going to called as the active site. Active site is a pocket-like structure. Pocket-like structure that is present in the enzyme. You can see a lock. Lock is having the key where the key is going to bind, right? That place is calling as the active site in the enzyme. Okay. So the active site is the pocket-like structure where the substrate is going to bind. Okay. Once the substrate is going to bind here, that is from the enzyme and substrate complex, then it is going to convert the product, and the again the enzyme is going to become a free here. Okay. And you have to remember, for every specific enzyme, the specific substrate will be there. Okay. For example, you can take amylase. The amylase enzyme is going to break down only carbohydrate. Okay. It is not going to break down the protein. Then coming to the trypsin. Trypsin is going to break down the only the protein, not any other substrate. Okay. All the enzymes are specific in nature. Okay. Fine. And you all know that the inorganic catalyst, for example, lead is going to be very active, and it will increase the rate of the reaction when the temperature is increases, right? But it is not case in the enzyme, because enzymes are made up of the protein. If you increase the temperature, then the enzyme is going to lose its tertiary activity, and the enzyme activity is going to be decreases. Okay. But there is only one enzyme is there that will be active in the high temperature also. That is nothing but the Taq polymerase. I think you studied that in the PCR. PCR, Taq polymerase. Polymerase, right? It will be active in the high temperature also. Okay, fine. Then coming to the enzyme function, activity, activation energy, guys. Mainly you have to remember here. This is the reaction, okay? There is a progress of the reaction and it is a potential energy. This diagram you have to remember very importantly. In absence of the enzyme, the activation energy is more, guys. What do you mean by activation energy? Means substrate is there, right? The substrate, the energy required to convert the substrate into the transition state. Okay. The substrate into the transition state, that is called as activation energy. Okay. If the enzyme is not there, means it requires the more activation energy to reach the transition state. Okay. So here you can see more activation energy required to reach for the transition state. If the enzyme is there, means it will take very less time. Okay. It is required the very less activation energy. Okay. You know that the enzyme is not going to be change the any product. Okay. With the enzymes also you are getting the same product only. Without the enzyme also same product only. But what is mainly going to change here is it is changing the activation energy or lowering the activation energy. That's it. Okay. So here you can see lowering the activation energy. It is not changing the equilibrium. It is not changing equilibrium of the reaction. Remember, equilibrium of reaction. It is only changing the activation energy, decreasing the activation energy. Remember. Okay. Here you can see here, guys. The substrate is in the higher stage, right? Substrate is in the higher stage and product is in the lower stage, right? Then the energy is going to be released. the energy is going to be released. So that's why it is called as a exothermic reaction. Exothermic reaction. The energy catalyzed, this reaction catalyzed by the enzyme is exothermic reaction. Okay. If the substrate is on the upper side and the product is on the lower side. If it is opposite, the product here and the substrate is here, means this is called as the endothermic. Endothermic. Okay. Endothermic reaction means it requires the energy. Remember this. This is very important. Okay, fine. As I already told you, the enzymes are having the tertiary protein structure and how they are going to react in the specific pH and temperature. Let us study here, guys. Okay. What you have to remember here is on the x-axis pH, on the y-axis enzyme activity. Remember, in case of the pH, for example, 1, 2, 3, 4, 5. Okay. For example, you have taken the 1A enzyme. Okay. This A enzyme is very active in the 3.5 pH. Okay. Till reaching the 3.5 pH, it is going to increase the reactions here. It is going to activity, enzyme activities goes on increasing. At specific 3.5 pH, it is reached the maximum activity. If your goes on increasing the pH means it is not maintaining its tertiary structure, the tertiary structure is going to break on down, and due to that the enzyme activity is going to decrease. So that's why you can see decreasing in the activity after the increasing the pH. Okay. The same thing happen in the temperature is also. Okay. On the x-axis temperature is there, on the y-axis enzyme activity is there. Okay. If your goes on, for example, this enzyme is active at the 40 temperature, 40. Okay. 40 temperature, the enzyme is active. The you can goes on increasing the temperature, the enzyme is becoming active. Okay. At the specific, at the 40 temperature it is reached means it is not going to active anymore. It is going to decrease its tertiary structure. Okay. The tertiary structure is going to lose. So that's why the enzyme activity is going to be decreases. Okay, guys. I think you understood this. Now we will go to this substrate concentration. Okay. On the x-axis, the substrate concentration is there. Okay. And on the y-axis, velocity of the reaction, velocity, not enzyme activity, velocity for the substrate concentration. Velocity means what? Speed, right? Speed of the reaction. If it goes on increasing the substrate concentration, the velocity of the speed of the reaction is goes on increasing. Okay. At particular period, it is going to reach the equilibrium. It is going to reach the equilibrium. Okay. It is goes on having the curve like structure. Okay. At equilibrium, it is not anymore going to increase. Okay. It will be equal. Okay. So where it is reaching the equilibrium, that is called as a Vmax, maximum velocity. Okay. And how do you have to maintain the, how do you going to be see the Km? Km is nothing but the Michaelis Menten. Michaelis Menten. Michaelis Menten. Vmax divided by 2. Vmax divided by 2. For example, now Vmax is 5. Okay. Vmax is 5. And divided by 2, how much it will come? 2.5. 2.5 is nothing but the Km. Okay. So this is about the substrate concentration, guys. Okay. Then coming to the inhibitor, if you don't understand means, once again I will explain this. Okay. At the end of the class. Then coming to the inhibitor, what do you mean by inhibitor means? For example, you can see, guys, you are the enzyme now. Okay. Your hands are the active site. Okay. Now you are preparing for the exam. Okay. Your hand should be filled with the book. Book is nothing but the substrate. Right. Now what will happen? You suddenly take a mobile in your hand. That is in the active site. Active side of your hand is now filled with your mobile. Okay, now you will take the mobile. Now, is that you are going to continue to see the mobile for about half an hour or for the 10 minutes? Now, you can tell me, is that book is going to come to your hand now? While you are while you are keeping the mobile in your hand, immediately the book will come to your hand. Is there a place for your book in your hand? Yes or no? No, right? That is nothing but the inhibitor, guys. The mobile come to your hand, right? That is going to inhibit. Now you tell me, you are active or not now? Are you going to be active when you're, this is the exam time. You are keeping your mobile in your hand. Okay. I'm not telling you that you are looking at the class now, but any other things you are looking. Okay. YouTube or something, reels and other things you are looking. Are you active now? No, right? The same kind of things will happen in the inhibitor also. The inhibitor is going to bind to the active site to the enzyme. Now the enzyme is not active because the substrate is not going to bind to the active site of the enzyme. Yes or no? So that's why it is we act. That's why that is called as a inhibition. Okay. So inhibition, you have to remember this very important thing, guys. The inhibitor mimics the substrate. Mimic the substrate means that is called as a competitive inhibitor. For example, you are taking the mobile only, but in that you are reading something, but it looks like reading only, but you are not reading. Okay. So that is looks like a competitive inhibitor. Here, what will happen? The substrate and the inhibitor look same. Okay. But when this inhibitor bind to the enzyme is not going to be active anymore. Okay. So that's why it is called as a competitive inhibitor. In the competitive inhibitor, inhibit looks same like the in substrate only. The example for this is succinate. Okay. The enzyme is succinate dehydrogenase. Instead of the succinate, a inhibitor malonate. Malonate is going to bind. Malonate. When the malonate is bind to the succinate dehydrogenase, the substrate is not going to bind. That is a succinate. Okay. So remember this is a very important question. Okay. And here you can see to the enzyme function, sorry, classification of the enzymes, guys. How can you remember this is oath lil. Oath lil. Oath lil. Oath lil means oxidoreductase. It will catalyze the oxidation and reduction. And the transferase enzyme, it will transfer the group rise. Example, you can see a kinase enzyme. Kinase enzyme is going to transfer the phosphate group, right? Phosphate group. We have seen in the glycolysis. The glucose is converted into the glucose 6 phosphate by the enzyme kinase, right? Here the kinase is going to be act as a transferase enzyme. Hydrolyse enzyme catalyze the breakdown of the ester, ether and the peptide and the pion bond. Mainly we can see in the digestion. Hydrolyse enzyme require the water. Then coming to the lyase. Lyase also break down the bond, but they don't require any water. Okay. But hydrolyse require the water for the breakdown. Okay. Then coming to the lyase, catalyze the removal of the group. Then coming to the isomerase enzyme, these are helpful in the transferring the groups, interconversion. Okay. For example, you can see epimerase and this is trans isomerase. Mainly they will do the shifting. Okay. Then coming to the ligase enzyme, you know that it will act as a glue and it will it will ligate the bonds between the bonds. Here you can see the enzyme which catalyze the joining of CO bond, CS bond, CN bond and PO bond. Okay. So these all are the example. Carboxylase, these all are the example. Kindly remember this. Okay. Then coming to the enzymes. In the enzyme, there will be two types, guys. One is the, 1 minute, sorry. Simple enzyme. Okay. Simple enzyme. The simple enzymes is mainly made up of the only protein. Okay. Then coming to the conjugated enzyme. Conjugated enzyme, these are made up of the protein and also the along with that cofactor. Okay, cofactor. If it is only protein is there means it is called as a apoenzyme. Apoenzyme means it does not contain any cofactor. It consisting of only protein. If the enzyme that consisting of the apoenzyme plus cofactor, then this is going to be called as holoenzyme. Holoenzyme, complete enzyme. Apoenzyme plus cofactor, it is called as the holoenzyme. Okay. And in there are three types, guys. A prosthetic group. Prosthetic group is a tightly attached. Okay. The example is peroxidase. Peroxidase and catalase. Here it is tightly attached. The group is heme group. Okay. Then coenzyme are loosely attached. Example, the vitamins NAD and NADP is acting as the coenzyme. Then metal ions, cofactors are metal ions. Example, zinc. Zinc is important, guys. Remember, zinc is acting as a cofactor for carboxypeptidase. Remember, zinc is acting as the cofactor for carboxypeptidase. And heme group is a prosthetic group in peroxidase and catalase. Important. Okay, let us solve some of the questions, guys. The ligases. Ligases is a class of enzyme responsible for catalyzing the linking, linking together of the two compounds. Ligase. Ligase will act as a glue, right? Which will link in the two bonds, which is not catalyzed by the ligase. Which is not catalyzed by the ligase, guys? Sai Charan, can you tell me which one I have to repeat? The correct answer for this, guys, is the option D, CN bond. CN bond is not going to be catalyzed by the ligase. Okay. Then here is another question. Which one of the following is not a fatty acid? Which one is not the fatty acid? Which one is not the fatty acid? Go through this. Which one is not acting as a fatty acid, guys? Just I will solve. I will not going to launch the poll, okay? Because we have a less time. A. You can see here, guys. Option C is the correct answer. Fatty acid, not a fatty acid they are asking. Right? Huh? Arachidonic acid is a fatty acid. Palmitic acid is a fatty acid. Okay? Then glutamic acid is not a fatty acid. It is a amino acid. Aspartic acid is the amino acid. Lecithin. Lecithin is also the amino. It is lecithin is a phospholipid. It is not the fatty acid. Okay? Remember only arachidonic acid and palmitic acid is the fatty acid. The correct answer is, you can see here, ADE. ADE is the correct answer. Lecithin is a small molecular weight organic compound found in the living tissue. Is an example of. Is an example of. Very good, Sai Charan. Very good. Answer me, guys. Which is the correct answer? Very good, Nukraj. Very good. Nita, can you go once again? Lecithin, we are talking about. Very good, Sai Charan. Very good, Krupa. Very good, Shruti. Very good, Pa. Now only we studied, right? Lecithin is nothing but the phospholipid. Right? Okay. The cofactor of the enzyme. Now only we studied carboxypeptidase. The cofactor of the carboxypeptidase is. I'm relaunching the poll. You can answer me in the poll, guys. Carboxypeptidase. Very good. Very good. Awesome, guys. Yes. Yeah. I'm sharing the result. No, not an issue. Okay. Zinc. Zinc carboxypeptidase cofactor. And here is the question, guys. Inhibition of the succinate dehydrogenase enzyme by the malonate is example of. It asked in the NEET 2024. Which is the correct answer? Very good. Option C. Very good, Nukraj. Very good, Shruti. Very good, Sai Charan. Very good. Yes. Competitive inhibition. Right. Okay. Here is another question, guys. Cellulose does not form blue color with iodine. Why? Cellulose does not form the blue color with iodine, whereas only the starch will form. Why? Why? Because the cellulose does not contain a helical structure. Very good, very good. I'm ending the poll. 80% have given the correct answer. Very good, guys. Because it does not have a helical structure, right? That's why correct answer is option B. Very good. Okay, this is another question, guys. Statement 1 and statement 2. Statement 1, low temperature preserves the enzyme in the temporary inactive state, whereas high temperature destroy the enzyme activity because proteins are denatured by heat. Option statement 2, when an inhibitor closely resembles the substrate in its molecular structure and inhibits the activity of the enzyme is known as competitive inhibitor. Which is the correct answer, guys? Very good, very good, very good, very good. Yes, Nita, yes, Nuti, yes, Supraju, yes, Simran. Correct answer is option D. Both the statement are correct, right? So that's why we will keep the vegetables in the fridge, because in the vegetables will in the fridge it will keep that in the inactive stage, right? If the more temperature means it will denature. You can go to this question also, guys, and I will launch the poll. Okay, first read the question, then I will launch the poll. Okay. The statement one, the protein is imagined as a line. Okay, so if protein is imagined as a line. This is the first amino acid. This is the second amino acid. Okay, the left end. Okay, this is the left end. Okay, the left end is the first amino acid where it is having the C terminal, whereas the second end, the last end, the right hand, that is having the last amino acid that is having the N terminal. Whether it is correct or wrong, it is wrong, because the first amino acid is having the amino terminal and the last amino acid is having the carboxyl terminal. The statement 1 is wrong. Statement 2, adult human hemoglobin having the 4 subunits, alpha and 2 beta. That is correct, right? The correct answer is option C. Statement 1 is false, statement 2 is true. Option C. Okay, guys, I will launch the poll. Students, I will tell you, whichever the chapter you're feeling difficult, okay, so that chapters you have to read once again. Okay, you have to read once or twice, thrice, then only it will become easy for you. Okay, you have to read that chapter, which chapter you're feeling difficulty, give the time for that chapter where you're feeling difficulty, read that chapter correctly, solve more MCQs on that chapter, which chapter is feeling difficult for you, right? Solve more MCQs on that chapter, then only you will become perfect. Okay, that is very, very important, guys. Okay, how you felt about this chapter, guys? How you felt about this chapter? This chapter you already studied, right? Yes, Patini, I will add you. Okay, fine. Okay, guys. Okay, thank you so much for the today's class. I think we are running out of the time. Kindly read this chapter once again, and this chapter PPT will be available. You can solve these questions. Okay, thank you so much. Take care. We'll meet in the next class.